Okay. All right. Good afternoon, guys. And Kennedy, please keep an eye out for anybody entering.
Okay.
If you see
Now ready?
Austin Newman, you can leave him in the waiting room for a bit.
Okay.
All right. Happy Tuesday, everybody. It is our last virtual biotech infusion. Um, Hope you guys have learned a lot over the past 4 weeks. Well, 3 weeks before now, and then we'll have a great session tonight running our gel, which you guys can see already set up on Cynthia and Trisha's cameras. So hopefully, if you're able to do the lab today, you kind of have out a very similar set of tools. And we will get started on that right after our speaker. So the main thing I wanted to chat with you guys about before our speaker is the scavenger hunt that I see a lot of them came in. Just want to see kind of what you guys found in your schools. If you have any fun stories about things you had no idea were in your building, but you found them. Anything you want to share? Maybe you found absolutely nothing, and that's terrifying, and now you have a growing wish list of things. Anything you guys want to share about it? Either if you want to put it in the chat or if you want to shout it out. Crystal's smiling. Maybe she's got something to share.
I just knew where most of it was because I have most of it in my room. Yeah, lots of grants, lots of grants.
Amazing. Yeah, so if you guys need help writing grants, look to Crystal. Awesome. Yeah, and Cynthia put a list of everything that you guys will need in the chat. So you can get those corralled if you need to. Did anybody go in someone else's closet and find something exciting that they didn't know was there? So when I was teaching, I was at Duluth High School and there are like 6 buildings. So I went to the other building and one of the bio teachers was like, I don't know why this is all in my closet. Here, take it. And handed me boxes of biorad stuff. She had an entire gel electrophoresis set, micropipettes, like a hot bath. And she's like, I don't need these things. And yeah, so I did for AP Bio. So I was happy to have found that in another stock room. So I do know like it happens that things get shuffled around. They get, hidden in corners. I bet Gretchen's got something hidden in one of those corners that maybe people haven't seen in a little while, but could get used for something fun. So.
I have to say, as AP biology teacher, I think my two most valuable pieces of equipment are the electronic balances and the hot water baths, because trying to get water to a specific temperature on a hot plate is practically impossible.
Oh yeah.
I hate doing that.
Yeah, keeping it anywhere near what you need. Yep. Yeah, you need a hot water bath. So fun fact, you can use the sous vide as a DIY hot water bath. So you can get one of those sous vide machines and it will keep a temperature. So you just pick whatever bin you want and put it in there with some water and boom, instant water bath. Digital balances, my favorite ones, I got on Amazon. If you just type in kitchen scale 0.01 grams, they give you some pretty good ones for about $15. So that is all we use at the depot. When we send out loaner equipment, that's what we send out. Yeah, $10. Yeah, it's Prime Big Deal days or whatever right now. So you could maybe go look on there and find a nice kitchen scale. So any other thoughts from you guys on anything you found doing scavenger hunt or anything interesting since last week?
Hey, this is Wayne. How you doing?
Hey, Wayne.
Good, good, good. Hey,
How you
you're
doing?
the Sigma with the dreads?
Yes.
Oh, that wasn't to us. Okay. All right. I just muted whoever was making noise. Oh, good. Okay, Cynthia put a link in the chat. Yeah. Uh, Zoom does not have the nice shorten hyperlink or create a hyperlink.
My apologies, guys. Hey, everybody. My bad.
Hey, good now?
Yes, ma'am. There's always I got to handle some
All
between
the things.
real estate teaching and everything else I do.
That's a lot.
Yes, ma'am.
Did you do a scavenger hunt? Find
I did
anything
not.
good?
No, man. I did not get a chance to do it. Just the honest to God truth. I lost a family member, so been trying to
Oh,
handle that, helping make funeral arrangements and
um,
all that kind of stuff. So that's where we've been at. So just always one thing after another, but it's all good. I'll get it done.
sorry about that. Thoughts
Thank
with
you.
y'all.
Thank you.
Yeah, I'm not going to take the link down. So if you want to go back and complete the scavenger hunt later when you have a chance, everybody's welcome to do that.
We definitely will.
Yeah, thank you.
Thank you.
Any other thoughts about the scavenger hunt? Any questions about the scavenger hunt?
No questions, but I did realize that a lot of the stuff that was on that list that we do not have, but we did have an incubator, which I worked to get through a grant, so I was happy about that.
Yay! Gotta love it. Well, and now you have some maybe a little checklist for your next grants. Some items to look out for. Awesome. Okay. Just got an email from our speaker, so he will be joining us. Let's see. So after this, I will not see you guys again until January. In January, we will be meeting in person on January 23rd and 24th at Central Georgia Technical College, Macon. So I hope that you guys can all make it. If you are having trouble getting a substitute for that Friday, if your school's saying they're not going to pay for it, we can help with that. The onus is going to be on them though. So you can let them know that they can email us an invoice for the substitute fee for you for that Friday, and we will reimburse the school for it. So that is up to your bookkeeper, whoever that might be. It might be another teacher in some cases, but it is up to them to email us to get that reimbursement. So we're happy to help. Just try to lower any barriers that we can to getting you guys to be able to participate and get what you need. So after the in-person training, you also you'll get a link from our bookkeeper about getting your stipend, and then you'll be in the system. So if you participate in any other workshops, we will just send the payments through that system. It's QuickBooks, so you can tie it to your personal email, and then it can kind of follow you around. So we try to provide a stipend whenever we can and give you guys some extra spending cash. It's not quick, unfortunately, but we will come through. So, and you guys are welcome at any point if you have questions about anything that we're doing. If you realize that after you've done this, you want to do one of the labs from this part of the training with your students this semester, just reach out. I did put the implementation plan in the Google Drive. There's now a folder called implementation off of the main drive. And it also has a spreadsheet for any of the labs that we do. It has a list of what the kind of consumables are that we would send, a list of the loaned equipment that we would send for that lab, and then anything extra that you would need. The extra things are like soap and water, paper towels, distilled water, that kind of stuff. So basically you can use that list, compare with your scavenger hunt list with what you already have in your classrooms. And if there's something that you don't have readily available, you put that on the list. On the implementation plan, it asks for what you need from us, That's where you put that information, and then we will work to get you those items by the time you want to do the lab. You are welcome to wait until after the in-person training. And then if you still want to do one from the fall, after we do the in-person training, you are welcome to do that as well. So happy to work with you guys however you want to fit in implementation plans with your students. Are there any questions on that? Awesome. Are we all doing good? Thumbs up? Yeah, surviving. Who's had fall break so far? Thumbs up? Yeah. No, I know. We're starting Friday. So, yeah, over the Indigenous Peoples break. So, I'm with you guys, just surviving till we
Megan,
get there.
will you send us any information about hotels that are available in that area where we're
I
going to be?
will.
Okay.
Yes. And I can do that sooner rather than later. Probably when I get back from fall break, I'll put that on my to-do list. But there are a couple good ones in Macon, kind of, I mean, good, it's good. But a couple that we like and recommend, so I'll send that list along. Just gonna
Are
follow.
we required to stay there, ma'am? Can we drive back and
No,
forth?
yeah, you're welcome to drive if you live close enough. So we usually aim to do regional training, so at least some people can drive to them from their home. So you are more than welcome to stay at home. I know I would rather sleep in my own bed if I can. So yeah, good question. All right. Yeah, Marissa's break is this week. Yeah. Thanks for joining us on your break. All right, I will go ahead and let our guest speaker in and let him make sure everything is working.
All right. Hey there, y'all.
Yeah,
Y'all here?
hey, Austin.
Hey, how's it going?
Good, how about you?
I'm doing really well today. Yeah.
Thank you. Thank you so much for joining us.
Of course. Yeah. Yeah.
Yeah, so you sent me what I think is the slides. Did you want me to share or are you able to share from your end?
I can share from my end.
Okay, great. All right, guys, we will transition to our speaker. So I would like to introduce Austin, who works at Takeda, and I will let him continue to tell you guys about himself.
All right. Howdy, y'all. Just want to be sure. Can y'all see my screen?
I see myself. Okay, there we go.
Okay.
There's
Awesome.
a PowerPoint.
Sounds good. Okay. I'm assuming I'm going to say present in Teams. Okay. All right. We're going to try that again. Okay. We're going to try presenting just on the computer then. Okay. Can y'all see that?
Yeah.
All right, awesome. So yeah, my name's Austin Newman. I'm a quality control supervisor at Takeda Pharmaceuticals. I work at the manufacturing facility for plasma. So we take plasma that's donated from individuals. We fractionate that plasma into its distinct parts, albumin and immunoglobulin specifically. And then we take that and we release it to the public for use, and typically in individuals. And we'll talk a little bit about the drugs that we make at this specific site. But yeah, so a little bit about myself. I'm a proud super nerd, cat dad, married. I love reading. Here's some of what I'm reading. I know y'all talked in the email you sent me about some of y'all are physics teachers, chemistry, biologists. Just want to let y'all know all of that is my gambit. I love that stuff. So another thing, I'm a lifelong learner, looking to go back for my PhD at some point. I'd love to get it in virology specifically. And that kind of brings me into some of my previous roles. So right now, I've worked at this specific site since February 17th, however long that is. And so I'm a little bit new to this area. Pharmaceuticals is kind of an exciting new adventure for me. A lot of my time previously was spent in the academic arena. So I was a laboratory manager and supervisor across 3 different laboratories. The funnest, I would say, was the Kraus de Kloot lab. There we studied cardiovascular neurobiology, looking specifically at hypertension, and then ways that we can modulate hypertension and obesity. In the Mukesh Kumar lab, we studied flaviviruses. So think like West Nile virus, and coronaviruses. A lot of what we studied was BSL-3. So when you think BSL-3, think the guys in the labs who had like the super fancy suits, and they had like super sterile labs. It is very much like that. So I enjoyed that a lot. It was very fun. We also had a BSL-2 arena where we studied what we call knockdown versions of coronavirus. So these are weakened versions of coronavirus that have specific segments of its pathogenic genomic array removed, which allowed us to test it in a BSL-2 setting. And then we also did molecular assay chemistry. This was a different sect of what we tested, but what we looked at was ways to detect viruses without necessarily needing super complex antibody assays. Think ELISA, right? So we would look at ways to use simple chemicals to react. God, it's been so long since I've done this work. Color compounds. That's about the best I can remember. They had a specific name for them, but yeah, that was the chemistry side. Again, I'm very heavily into biology, and so yeah. Then with couple of years ago, I worked with Jessica Carter. All of this research here, this work occurred at Georgia State. I highly recommend Georgia State for any research. If you have, students who work with you who are interested in research, I would say that of the public universities that I've worked at, for biology specifically, it's hard to beat Augusta University and Georgia State University. Emory comes pretty close, but I worked over there for a while. I'm going to be real with you. If y'all are in Georgia, Emory can be grueling. They have really high expectations from their students. Some of those students that I worked with over there were working sometimes 12-hour days in the lab, which is kind of unacceptable, and then being paid a very meager salary. So prior to all of my management and supervisor experience, I had 4 years on the benchtop. I worked across a couple labs where we did endocrinology, vascular biology, epigenetics, microbiology. I've worked with ferrets, macaques, rhesus, a lot of mouse work, a lot of mouse surgery. So the Takeda Pharmaceuticals Covington manufacturing site produces 2 products. That will be expanded to 4, but when I say 4, it's kind of 3 of the same product, 1 of the different. So we produce albumin in a 5, 20, and 25% matrix. We also produce immunoglobulin G, known as Gamma Guard, and both of these are IV drugs. So albumin is what we call an osmolyte. It actually increases the oncotic pressure inside of the body. For every gram of albumin you have, you can usually hold on to 18 grams of water. So we use albumin in settings where we have individuals who are in septic shock or who have had massive burns across their body to help their body hold on to water and keep them from going into hypovolemic shock. Hypovolemic shock specifically being when you lose too much blood volume, okay? Immunoglobulin G is, or our Gamma Guard product, is used in individuals with primary immunodeficiency syndromes. So immunoglobulin G is the most common immunoglobulin produced in your body. When you're exposed to any kind of viral or bacterial infection, you will produce an immunoglobulin. That immunoglobulin has specificity to whatever it is that it was produced against. You have a large number of these proteins circulating in your blood at any given point in time. We take them out and give them to individuals who, let's say, have primary immune deficiency or SIDS, severe immunodeficiency, and then they can actually fend off viruses, bacteria for about 21 days after we give that drug to them. So it's an absolute lifesaver for anyone who has an immune dysfunction disorder. And then we're located out in the middle of nowhere. I got to be honest with you. It was a heck of a drive. I used to live in Atlanta, and that was about a 45-minute drive there, and then an hour and 50-minute drive back. I loathed it. So specifically, though, I work in the quality control lab. We have a lot of different sites or areas across our specific site. So I'll talk a little bit about those areas. But what our lab does is while we're making the actual product, we receive samples from the in-process creation. We then test those samples for specific constituent parts, wherein, like, we're looking at sodium, we're looking at the pH of the sample, we're looking at the total protein of the sample. We have a really unique way of studying total protein, actually. We use a system known as LECO, the LECO FP628. It basically takes a dried protein sample, drops it into a crucible at 950 degrees Celsius, incinerates it, reacts whatever burns off in the incineration process with pure oxygen, and then it's basically a fancy mass spec after that. It reads whatever reacts, and then it tells you a percentage of what nitrogen yield you have from what it reads. And that'll give you a good idea of what your total protein is. So we have actually a couple really interesting methods used in our laboratory. So I talked a little bit about LECO. Obviously, every single chemistry laboratory in any pharmaceutical site that you go to is going to have gas chromatography. It's a given. We use liquid, we use mass spec, a lot of ICP. So if you haven't heard of ICP, it's ion coupled plasma. It actually uses a plasma array to heat up the sample for mass spec. It's really interesting. It's extremely sensitive, and it's very useful for metals in specific. We onboarded these instruments because we thought when our site was set up about 14 years ago that we would be testing for heavy metals in blood. Turns out the FDA cared not at all about that. They wanted to know about kind of the lesser size metals. So they were really looking for basically just sodium and potassium. And so we ended up getting the ICP-MS. It's not the best tool for the job, so we are looking at going to flame photometry right now. We also use UV and absorbance assays. We use ELISA assays. That's a lot of our IG unit. So I'm over the albumin unit. Very specifically, I do albumin testing. One of my counterparts, Abby Lee, she does all of the IG testing. She's the supervisor for that group. But also something that we do is very basic assays. A lot of our testing is extremely basic. Think like TOC, total carbon, or total organic carbon. Think pipetting skills, balance usage, things of this nature. And genuinely, whenever we onboard new analysts, these are some of the things we notice are lacking from all of their years in education. So career paths at this site are varied, right? There's actually a ton of options out here. We employ 1,250 individuals at any given point in time. That number goes up and down naturally. But what I always tell people is if you're looking for something, you can typically find it at our facility. Our QA division, or our quality assurance division, is the one who, they're the people who are read up on the federal code. They're the ones who keep us compliant, who keep us from doing something that the FDA would say, oh, no, buddy. Quality control, that's the group that tests everything at the site. We're responsible for all of the testing at the site regarding any of the product. We also have, and this is pretty much ubiquitous throughout all of our production facilities, a lab excellence team. So our lab excellence teams are basically the Swiss Army knives of the unit. So if you have students who are super interested in being, a statistician or something related to that effect, a mathematician, someone who would know instrumentation inside and out, someone who would do a lot of data processing and data analytics, our lab excellence group is honestly fabulous for that. We also have manufacturing sciences. Now, manufacturing science is a little interesting because it's basically the academic branch of what you would consider pharma. This is where people are conducting novel testing to try to improve our manufacturing process. So the people that we employ in our MS unit are, at the end of the day, actual scientists. Most of these people come with very heavy science backgrounds. They have master's degrees, they have PhDs, they have 20 to 30 years of experience. And the newer individuals that we hire on in the manufacturing sciences unit are typically engineers, all right? Now, I've stated a lot of stuff here about these units that we have at our site, and it says bachelor's, master's, or PhD. When I was coming up through high school, I remember not every kid dreamed of going to college and taking on $50,000 of debt. So what I always tell people is we have something called the CUB unit, the Central Utilities Building. These are people who make somewhere in the range of $70,000 to $180,000 with nothing but a vocational school degree, a technical degree, and typically it's an HVAC, welding, plumbing, and electrical. These people are highly paid. They know their field, and it's honestly marvelous. They're the ones who keep the lights on for us, so they're the team that we rely on the most. We absolutely have to have them. They control everything at our site. CUB, just a really interesting fact for everyone here. At our site, we use a ton of denatured alcohol. Now, denatured alcohol is ethanol with a certain percentage of methanol seeded into it in order to keep people from drinking it, basically. Methanol makes you extremely sick. We keep about 600,000 liters of it at our site, and that number will go up to 900,000 in about 3 months. So this unit specifically, our CUB, our Critical Utilities Building Unit, these are the people who keep all of that on lockdown. Now, another option that we have at our site that's a really big thing that attracts a lot of new talent, straight out of high school even, and straight out of college, is our IT unit, which is constantly growing because one of the interesting facets of living in the middle of nowhere and being in the middle of nowhere as a site is that it's hard to get Wi-Fi. It's very hard to get Wi-Fi. So a lot of these teams that we have built up at our site focus on ways to decrease overall network usage and find ways to make us a little bit more efficient on our IT side. So something I wanted to go over, since you guys are all teachers and you're feeding minds every day, and you get to guide a lot of, I'm assuming y'all are middle and high school, so y'all get to guide a lot of impressionable human beings to go on and do something with their life. There are qualities that I seek when I'm hiring. I've hired probably 12 candidates now, or at least been a part of the hiring process for 12 candidates. So I seek great manners, answering questions completely, admitting ignorance. And then when it comes to resumes, I really seek people who have SMART goals on their resume. I don't want a rundown of where you worked or why you worked there or what you did when you were there. I want specific goals and things that you achieved when you were there. I decreased the turnaround time by 25%. I was able to complete this project in X amount of time. And things like that are super, super important on resumes. So when you're leading your students, I would say, drive home the importance of instituting SMART goals on their resumes, especially when they go into college. They should be focused on, or when they go into whatever they go into, they should be focused on delivering to the people they are seeking to get hired by their goals and what they did while they worked a job. Not necessarily that they worked a job, but that they actually did something when working that job. When I see actual numbers on paper for analysts, I immediately they immediately come up to about the top of my list in terms of hiring. I always say this, especially about everyone we hire. I can pretty much teach anyone how to run our assays. It does not take a college degree to run our assays. What the college degree does is it hones a person's skills and abilities. But there's also something a little bit deeper there. I need people who can come to me and show real desire to grow and learn. So having that mindset of saying, I want to be better, I want to grow, I want to learn more, I want to do more, taking initiative and things of that nature, these are things that I absolutely look for in individuals when they come to me and want to be hired. Another thing is, and this is something that we at Takeda really want to drive home, confidence during interviews. So whenever we have people who come to us, we're looking mostly for someone who sits straight up, can answer questions, doesn't hide their face, can have a really good conversation. And you would be shocked at how often we encounter people who kind of slouch down and put their elbows on the table and kind of ball up. We try to make it a very hard to describe. Interviews in general are always a little nerve wracking. But I would say specifically for us, we try to make it at least a little bit, laissez-faire, laid back. So If you guys, I know Megan offered, if you wanted to contact me, I'm always willing to help and give people opportunities and guide people on what we're looking for as a company. So you're always welcome to contact me. Best way is typically through email. So you can throw an email my way. So yeah, that's all I've got for y'all. If y'all have questions, I'm happy to have a conversation and speak. There was something else that I was willing to share with you guys if you wanted. So I did pull up, I want to be sure y'all can still see my screen.
Yep.
Okay, cool. So what I pulled up is what our jobs look like at Takeda. And so I went ahead and pulled up what the analytical chemistry hires for and what the job roles are about. If y'all see this as useful, I'm happy to kind of send that out to y'all, to Megan, and let y'all do whatever y'all would want with that. But this, I figured, might be useful for instructors who are looking to grow students toward a certain path, if you may. So anyway, that's all I've got.
Thanks, Austin.
Yeah, no problem.
Yeah, I think I saw some people nodding. So if you want to email that to me, they have a Google Drive going for the workshop. So I'll
Sweet.
put it in there. Thank you.
No problem.
So you can stop sharing.
Okay.
There we go. Awesome. Yeah, I saw a yes, please send it. Do you guys have any other questions for Austin? Feel free to shout them out or put them in the chat. I can read.
Not really a question, but I do appreciate the comment about like the SMART goals. I have my kids write resume and genetics because we do like a mock career fair. So like that's very helpful in knowing now what people are looking for on resumes. So if there's anything else that we can pass along to them, that's good because they don't really have an avenue to write those in a lot of classes anymore.
Yeah.
So that's just something we kind of integrate when we can. So that was great. That was good feedback.
Yeah. So I always tell people this is a very, very interesting practice. Take your goals, put them into an AI generator, have it write it up for you. I'm telling you right now, we receive at any given point in time through our contracting company, sometimes 2,000 resumes. They absolutely put them through an ATS AI reviewer. All right. When you have a resume that you build with AI, it builds it in such a way that it can pass an ATS reviewer. A lot of our resumes can immediately get thrown out, and we lose a lot of quality in our candidates because of this issue. This is something we've actually brought up. So our site is working to like move away from using AI, but I would say that having that clarity of language from an AI kind of writer is a lot more helpful.
Thanks,
He
Siri.
really had something to say about that. Yeah.
Yeah, and I think we had touched on it before, just the soft skills, because you can teach all the hard skills, teach
Yeah.
them your equipment, but if they don't have that just general background, and I forget what
Yeah.
Shelby called them, sparkly skills or something, but yeah, just being able to be there on time. And I know you guys work around the clock. Well, you might not, but there is somebody at Takeda round the clock.
Yeah.
So getting to your shift on time to relieve the last person is of utmost importance. And
Yeah. Yeah. I would say, timeliness, punctuality, those things are always in style for us as individuals who hire. And I want to drive it home, students should be, I hope that they have an inquisitiveness about them. They want to learn more. They have that desire to get out and know the world. I don't need you to be Einstein or anything of that nature. I just need you to want to go there with me, and that's what I look for. And I think that's what we look for at Takeda in general is just, we have hiring parameters that we go by and we look at specifically. And so we always try our best to find people with integrity, with the ability to be punctual, with the ability to work with others. Teamwork is a huge thing. on my shift specifically, I run a team of 16 people across 4 shifts. And we have a day shift, a night shift, and we go full blast. We're on 24/7, no matter the holiday. And so something that's super important are communication skills, people being able to work together in a team environment and communicate with one another effectively is integral. So seeing what we've seen so far, I would say out of the college graduates that I get, I usually try to hire college graduates. I'm not even going to lie there. They haven't been beaten down and torn apart by the professional world yet. And so I try my best to hire college graduates because they've got that gung-ho spirit about them. And so, yeah, it's always one of those things that I see kind of lacking in the individuals that we hire are those soft skills, you know?
Yeah. Awesome. Thank you so much.
Yeah, of course. Yeah. If there's ever anything I can do, y'all feel free to reach out to me. I'm here as a resource. I love education and academia. So if I can support teachers in any way, y'all got it.
That's great. Yeah, we're always trying to get you guys connected with teachers, and a lot of the teachers here are from the at least area around Covington, not Newton County specifically, I think. But
I'm so sorry.
yeah,
Maybe y'all like it. I don't know. We just moved out here. It is very rural.
well, you and Gabe, so you've got good company at least.
Yeah.
But yeah, well, yeah, we'll try to send students out your way. They're at
Awesome.
least within commuting distance, so
Sweet. Okay.
yeah.
Awesome. I appreciate y'all's time today.
Yeah, thank you, Austin.
Of
We
course.
really appreciate your time.
Thank
Thank
you.
you so much. Thank you all.
Have a great one. Bye, Austin.
Bye-bye.
Awesome. Yeah, so he was actually a last minute ad. So I'm really glad that Takeda pulled through and got us a speaker for today. So hopefully you guys found that insightful, and hopefully our 4 speakers have given you guys a pretty kind of, I feel like a well-rounded view of kind of what's available in life sciences, and how your students can work in life sciences even if they're not a science-y person necessarily, if they don't consider themselves science-y. There are still positions for them to be out there healing and feeding and fueling the world. So, are you guys ready to do the lab?
Yes.
And I guess maybe that's
We're
Cynthia
ready.
and Trisha.
Yes, 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 yes.
Awesome. Okay, so I am gonna sit here and create the breakout rooms like I, again, should have been doing earlier, but it's fine. This is fine. I might be putting these in the right. It seems like they're already grouping you guys. Or maybe I need to, okay. Let's go back. Maybe I picked the wrong thing. Recreate. Let participants choose. That's it. Okay. All right. That looks better. Okay. All right. First room is Trisha. Second room is Cynthia. And I'm opening the rooms. So you guys should be able to join now yourself, but if you need help joining, I will hang around and can put you in your room.
Okay, Megan, I'm gonna go to Cynthia's room to record.
Okay. Yep. Thanks, Kennedy.
You're welcome.
Megan,
Hey, June.
hey, can you put me in my room?
Yeah, whose room is it? Which room, Cynthia or Trisha?
Don't know. I can't see.
Okay. It's gonna take me a minute.
Okay.
Because I don't have that open right now. Vicky, do you know where you're going? You're muted.
I'll be going in Trisha.
Oh, I see it.
I'm not
Okay.
able to see the rooms option.
Okay.
Missing.
I can't see them either.
Okay,
Yes.
Shakira, which room are you going to?
Miss Trisha's room.
Trisha's. Okay. And Miss Lewis, whose room are you going to?
Oh God, Cynthia,
Okay, there you go.
thank you.
You're welcome. All right, Pam, do you need help?
I just got home, so I'm going to transition to my computer
Okay.
and then go into a room.